So thanks again, Walter. Thank you, David. And so I want to tell you, so it turns out I started my position at Case Western exactly 30 years ago today. That was just by chance, but it turns out that's exactly when my job started at Case. And so I want to tell you a little bit about the lab during these 30 years. And so first, though, let me say a few words about Richard Armstrong. So Richard, I did consider him to be a mentor very much for me. I served with him 13 years with him as an associate editor while he was editor-in-chief. And I learned a wealth of things from Richard, especially from the trips we used to take to Washington for the editors' meeting. Sort of watching him operate was extremely instructive and edifying. And so I've always been grateful for the opportunity to work with him. So, and for those of you who don't know Richard, I did want to give you a little bit of insight into his personality. So I had always been told that Richard loved the Lange Motor Museum in South Nashville. And I had never been there till this last year. We had some friends in town and we were looking for something to do. So we went to the Lange Motor Museum. And I had always assumed that Richard must have liked the Lange Motor Museum because there would be elegant cars there. And Richard liked cars and he liked elegant cars. And so indeed, there are elegant cars at the Lange Motor Museum in Nashville. And I really enjoyed this museum. You're really encouraged to go there. But there was something else at the museum that made me realize, this is really why Richard liked this museum. And here is an example. So there are some cars there that are like comically bad ideas, like cars with propellers propelling them. And so I think this hit the nail on the head of why Richard would have liked this. It was this blend of elegance and the really comically bad idea that he would have taken great delight in both things. And so I really miss Richard a lot. I want to point out another connection I have with the Armstrongs in the laboratory setting. And that is that his daughter, Katie Armstrong, when she was a biology major at Rochester University, spent 2 summers in my lab doing lab research. And so this is Katie. I can't believe it was 10 years ago, but it was 10 years ago. So Walter referred to the mentoring philosophy document at my lab website. This is the link for it. And let me interject at this point. If anyone would like a copy of these slides, I know there are former members of my lab present. I put in the chat a link to the slide deck, and you're welcome to download it and have a copy for yourself. I'm not going to say much more about my mentoring philosophy, but I do want to note that I've made a huge number of mistakes as a mentor over the years, as well as a scientist. And so I hope that you keep that in mind as I give this talk, that I appreciate all the very positive things that Walter said. But I'm all too well aware of the fact that I'm an imperfect mentor. I've made many mistakes, and that's just part of being human, I guess. So I gave a practice talk earlier this week to my lab, and they said that I should tell you what the outline of the presentation is. So that's why I'm going to show you this. So we're going to go from the founding of the lab 30 years ago, all the way up linearly to a current project in the lab, our early stage drug discovery project that targets Charcot-Marie-Tooth disease. And so I'm going to take you, how did we get there from starting the lab? And then after I do that, that takes us to the present. I'm going to back up just a little bit and but just very briefly highlight the other 4 projects that we've had in the last 30 years, very briefly, and then give you some concluding comments. So that's the outline. It's not completely linear in terms of the chronology. So I've had many mentors, and I really view my own being mentored and continuing to be mentored as absolutely essential to whatever success I've had in science. And so I did my PhD with Ming-Dao Tsai at Ohio State. I probably would not be in science today if it were not for Ming-Dao. He knew exactly what to say to me to motivate me and to get the best out of me. This was on graduation day. Ming-Dao came over and hooded me, which I really appreciated. And then I went to Yale, of course, with Jim Prestigard. I can't believe that I don't have a single picture of me with Jim Prestigard during the Yale years. But he actually never changed with time. He looks the same today as he did then. And so this is a picture taken 7 years later of Jim and I at a conference together. Jim also was a wonderful mentor, and I'm extremely grateful to have had the opportunity to work with him. So I took my first position at Case Western Reserve in the Department of Physiology and Biophysics. And my chairman there who recruited me was Tony Scarpa. And so this is a picture of Tony. And Tony also was a mentor. One would never call Tony necessarily a warm and fuzzy person, but I really learned a lot of stuff from Tony. He was a wonderful chairperson. Those of you who've never heard of Tony Scarpa will know him nevertheless. So after he left Case, he became the director of the Center of Scientific Review at NIH. And Tony is the person who changed the length of NIH grants applications from 25 pages to 12 pages. So a great many of us who are listening in on this talk have Tony saved us hundreds of hours by reducing the length of the grants, both in terms of writing and reviewing those grants. So, so I showed up in Cleveland in 1991 with my family, and I immediately then needed to set up a lab. And so I needed someone to work in my lab. And so I met a taxi driver named Jim Schwanek, who was driving taxi in Cleveland at the time. And it turns out Jim Schwanek had just graduated from MIT with a degree in physics, and taxi driver just happened to be the best job he could find. And so I quickly snatched him up into my lab. And so Jim was the Sanders lab for the first 2 years. And I think Jim published about 5 papers with me. He was a wonderful first person in the lab. I later had the pleasure of being the best man at his wedding. This is my wife, Becky, here, and obviously Jim's wife, Cheryl. And he eventually did leave the lab to move on. He got offer for getting paid 3 times more to work at UNICEF. And so that was his next job. So the first project we started on at Case Western was working on bicelles. So the bicelles project had started in Jim Prestigard's lab. And when I was in Jim Prestigard's lab, we developed the CHAPSO DMPC bicell system. And so bicelles are lipid bilayered discs that are edge stabilized by detergent. When I got to Case, we had really, at that time, very primitive, older NMR capabilities. And so I couldn't do like protein structure right away till we got new instrumentation. But they had an old instrument that I could do like bicell development work with. And I asked Jim Prestigard if it would be okay if I tried to develop a second bicell system based on the use of short chain PC as the detergent instead of CHAPSO. And Jim said, of course, as if it was sort of a silly question for me to ask him. But I've always appreciated that generosity, totally non-territorial. And I've always tried to pass on that same generosity, paying it forwards. So, so while I was at Case, Jim and I developed the DHPC, DMPC bicell system. And so bicelles have become sort of a major class of model membranes. If you do a Google search on bicelles, you get about 75,000 hits. One of their properties is that certain compositions will line up with a magnetic field, which is sort of an interesting property. Now, bicelles have taken on a life of their own. There's sort of a small community of people who ended up working on them. In this community was my friend Scott Prosser. So Scott Prosser began to work on bicelles several years later and got a job at Kent State University, which was right down the road from Case. And so we became friends and collaborated some, and over the years have shared many scientific ideas together. This is Scott in China. These are cormorant fisher birds. And so originally, you know, the bicelles were thought to be bilayered discs. Scott and other investigators over the years showed that it's more complicated than that. That depending on the exact detergent to lipid ratio, bicelles can be either Swiss cheese, intermediate structures, bilayered discs. And if you add enough detergent, they're not bicelles anymore, they're classical mixed bicelles. And all of these different mixtures are potentially useful as model membrane systems. So over the years, my lab's main has continued to come back to bicelles from time to time with various technical innovations. And I list them here. Don't try to read them all. I'll just give you one of the things we did was I actually named bicelles originally in 1995 and published the name, and that it did catch on. And then very recently, we've developed a new class of bicelles that have a lipid raft-like composition. And so here's a depiction of one of these. So it's got both sphingomyelin and cholesterol in it and other lipids. And so this is the first isotropic model membrane system that mimics lipid rafts. And so this work was started by Haley Harrington and Kate Mittendorf in my lab. One year, Kate dressed up as me for Halloween, and actually, I think did a pretty good job at it. She even has my cheapo Timex watch here. The one thing she didn't quite get right was the hair. Thank heaven she didn't get that right. And so anyway, so they started this project, and then James Hutchison came in and really finished the characterization and early applications of these. James recruited a whole boatload of off-campus collaborators who contributed to this recent JAX paper. And one of the things that we did was we actually took the first cryo-EM images where you can actually see bicelles. And that's because these new bicelles persist at very low concentrations. So anyways, we've done a lot in the years with bicelles. Thinking more about technical innovations that my lab has contributed over the years, another thing we've worked on are these are amphipathic polymers known as Amphipols. And these were first conceived by Jean-Luc Popeau in France, and he developed early systems. In around 2000, my lab worked with Anatrace, a detergent company, to develop a different class, what we thought was an improved class of Amphipols called the P-mol series. And Amphipols can form complexes with membrane proteins to solubilize them. That's what they were originally developed for. Now, not all that much has been done with these polymers over the years, although there are a few cryo-EM structures now done using the P-mol series. But very recently, another James Hutchison project in collaboration with Riccardo Caponi was to actually take the envelope protein of the SARS coronavirus, and this is a membrane protein. And what they did was they purified the protein in this P-mol Amphipol mixture to solubilize it. But then what they found was they could add the mixtures to living mammalian cells. And here on the right is the time-lapse series. The protein is green, and you can see that this protein from SARS actually inserts into the membrane of these cells and traffics to the trans-Golgi network. And so it's remarkable that Amphipols can actually be used as a delivery agent to facilitate insertion of membrane proteins into cells. We had done this a little bit when these were first developed, but only with model membrane systems. And so this was sort of an interesting observation. There may be implications for SARS, but more importantly, it suggests that perhaps Amphipols may enable the possibility of using membrane proteins as biological therapeutic agents. Now we have a way to deliver them to cells, into the membrane of cells. And so that's sort of a new thing. So we've done a number of other methods development projects over the years. These are some of the folks who have done these projects. I won't go through all of them. I'll note a couple. So Olga Vinogradova was my first graduate student. She's now on the faculty at UConn. And Olga was really a pioneer in early exploration of NMR conditions for applying them to very large membrane proteins. A wonderful student to work with. And a second project, which I always thought was sort of a fun project, was by Lorenzo Olive, an undergrad in the lab. And what he developed was a E. coli medium that when you grow your cells, when it's time to induce the culture changes color. So you don't have to measure A260. You can just look at the medium. When it changes color, you add IPTG. That never really caught on in the broader community, but I always thought that was sort of a cute paper. So anyways, these are some of the folks that have done methods development in the labs over the years. So back to Case Wesker. So midway through my 11 years in physiology at Case Western, I realized that I didn't understand what any of my colleagues were doing because I'd never taken a course in biology since high school. And so I thought, I really should try to figure out what it is my colleagues are working on. So I bought a copy of Molecular Biology of the Cell and read it. So that's how I got familiar for the first time with, oh, that's what endoplasmic reticulum is. So around 1995, here in the top left is my lab at Case Western Reserve University and the folks that were in it. And we began a project that combined both my PhD work in enzymology and my postdoctoral studies in membrane systems by looking at a membrane enzyme, E. coli diacylglycerol kinase. And our studies of this protein were enabled by the gift of Jim Bowie to my lab of a very excellent overexpression vector for this protein. And once again, Jim's generosity led not only to a long friendship, but it also led me to conclude that anytime anyone ever asked our lab for a reagent, we would give out the reagent, no questions asked, and we would provide all the documentation we possibly could to make sure that they could use what we sent them. And so that was a Jim Bowie legacy to the lab. And then around the same time as this group, I recruited Bonnie Gorzel as my lab manager there at Case Western. So in our early studies of diacylglycerol kinase, we were very interested in understanding how does a membrane kinase differ from a water-soluble kinase. And we came through a number of studies, we came to the conclusion that a membrane kinase is basically evolutionarily limited by its environment, so that it's very hard for it to evolve out of its own special biological niche. And in fact, it's not even necessarily a really good enzyme chemically because of its biological niche. And yet, nevertheless, it's very successful in its membrane niche. And we liken this to diacylglycerol kinase being the world's greatest third grader. And the world's greatest third grader would be little orphan Annie. And the reason she's the world's greatest third grader is because she's been trapped in the third grade for over 80 years. So if you're in the third grade for 80 years, you would be a really good third grader. And so that's sort of what it was like for diacylglycerol kinase. It's trapped in the membrane. It can do what it needs to do in the membrane, but because it's in the membrane, it's hard for it to evolve into a powerful family of membrane protein kinases, the way the water-soluble kinases have. So another thing that we set out to do with diacylglycerol kinase was to determine its three-dimensional structure using NMR methods. And so a number of people contributed to this project. It really was started by Olga first, but then carried on by Kirill Oksenoy, a fine postdoc in the lab, under the guidance of my next door neighbor at Case Western, Frank Sonicson. And so this is Frank on a ship in the Baltic a number of years ago near his home in Kiel. And Frank is like a master NMR spectroscopist, but he has a special talent for training other people in NMR. And when I was next door to his lab, that was unparalleled opportunity for people like Kirill to be trained in NMR methods and the related computational methods. Kirill started the project. It was finished by Hak Jun Kim and Wade Van Horn especially. And after really 15 years of working on this project, we did determine the structure of diacylglycerol kinase. We were able to get it published in Science. And so here it is. And so this was a fairly notable technical achievement in NMR spectroscopy. And there were other people who did membrane protein structures around the same period of time, but I believe this was the first one where there was not first a crystal structure that had been solved prior to the NMR structure. So this was sort of new from that standpoint. And it's also just a fairly large and complicated membrane protein. So we were really thrilled about this, but we were much less thrilled when 2 years later, Martin Caffrey and his postdoc, Dien Van Lee, published the X-ray crystallography structure of diacylglycerol kinase, shown here, which it turns out is a different structure than the NMR structure. And so Martin had very politely told me all about this well in advance of the Nature publication. So we had a chance to look into this to figure out what the basis for the discrepancy was. And the problem is that it turns out that the structure we had determined, which we determined in dodecyl-phosphocholine micelles at 45 degrees Celsius, is a misfolded structure. The enzyme is not actually active at 45 degrees. Now, you may ask, how could we not know this? And the answer is Chuck Sanders made a mistake. So early on, when we were doing NMR structural studies of DHEK, I measured the activity of the enzyme in DPC micelles at 30 degrees Celsius, where it is active. But we had never tested whether at the NMR temperature of 45 degrees it was active until after the crystal structure came out. I went back and I did those experiments with my own two hands. And when you do the assay at 45 degrees, you can watch the activity die. And so that's because the protein is misfolding into this incorrectly folded state. So I've always been very disappointed with myself about this. It was my fault. Nevertheless, it remained an impressive technical achievement for the members of the lab that they were able to do this. And so science goes on. But I think the lesson of the story here is you can never pay too much attention to detail. You really have to pay attention to detail, or sometimes you really get burned. So another thing we did with diacylglycerol kinase was we had a library of single cysteine mutants that we had gotten from Jim Bowie. And we did many, many experiments on all these different mutants. I won't go into the gory details, but the most notable thing that came out of this was our, I wouldn't call it discovery, but our annoyance that a great many of these single cysteine mutants are highly prone to misfold to form incorrectly folded structures. And this was initially viewed as a terrible nuisance. But about that time, I was beginning to realize that almost nobody in the world was interested in diacylglycerol kinase, and that I should find some new projects that people were interested in. And it turns out that I also quickly learned that there are a number of diseases related to mutations in membrane proteins that cause them to misfold. So at that point, then I decided we're going to embrace misfolding and try to understand misfolding in diacylglycerol kinase as a model system for misfolding in human proteins that causes disease. And so I had a really outstanding student, Joanna Nagy, who published, I think, about 7 papers with me. And Joanna eventually was succeeded on this project by Debbie Mee, that many of you here at Vanderbilt know because she still works in the high-throughput screening facility. And so Joanna took up this problem. And what Joanna showed was that she was able to basically assess the thermodynamic stability of these different diacylglycerol kinase mutants. And that stability reflects the equilibrium between the folded form and the unfolded form. And then she also had an assay for quantitating misfolding. And what she was able to show was something that had been seen previously for water-soluble proteins, but never for a membrane protein. And that is that mutations that destabilize a membrane protein make it prone to form incorrectly folded aggregates or misfolds. And so this, I thought, was a very, very important paper. I still think this is one of probably my top 5 most important papers in my career. And it led us then to think about going after this in a human membrane protein. Do we see the same trends in human membrane proteins? And so Jeff Myers and I wrote a review on disease-related misfolding of membrane proteins around the same era. And I also looked for a new project, a human membrane protein for which mutations are known to cause human disease. And I wanted to do a protein that other people weren't already studying. So there were a number of proteins that do misfold and cause disease, things like occur in retinitis pigmentosa with rhodopsin and cystic fibrosis with the CFTR protein. And I didn't want to do what other people were doing. So eventually we stumbled on this obscure membrane protein, peripheral myelin protein 22. And mutations in this protein cause a disease called Charcot-Marie-Tooth disease. So it turns out Charcot-Marie-Tooth disease was in the news this week because country music legend Alan Jackson, who's giving a big concert here later this year in Nashville, has been diagnosed as having Charcot-Marie-Tooth disease. He's going to try to continue his career despite the fact that this can be a debilitating disease. So we decided to take up peripheral myelin protein 22. So now about this same time, this pre-review that we published was published in 2001. In 2002, I moved to Vanderbilt, which was a wonderful move for me, although I do miss, there were a lot of great things at Case that I missed, especially friends there. And the reason, and we basically decided to, and so when I moved to Vanderbilt, I needed to repopulate my lab. Only one person came with me from Case to Vanderbilt. Kirill Oksanoy came for 1 year. And so the first thing I did was the best thing I ever did in my career, which was to hire Irina Hadji-Slimovich. So Laura Busenlaner was a student at Texas A&M who I contacted then about coming to my lab to do a postdoc. And Laura said, no, I can't come to your lab because I'm going to Richard Armstrong's lab. But I have this friend in California who's looking for a change of venue. Her name's Irina. So anyways, the rest is history. A few months later, we recruited Irina to my lab to be my lab manager. She's been a trusty, reliable, outstanding lab manager over the years. This is a picture of her with my wife, and I'll explain the outfits later. And of course, the move to Vanderbilt is thanks to Walter Chazen and Mike Waterman. Mike I knew from study section. Walter and I met on a bus in Japan and had a wonderful 2-hour conversation on the way to a conference that led me to being recruited eventually to Vanderbilt. So anyway, so when we got to Vanderbilt, we started working on this peripheral myelin protein 22 project. The protein has 4 transmembrane spans. Mutations at the blue sites cause this disease, Charcot-Marie-Tooth disease. Jeff Myers and Chuck Mobley spent 5 years basically figuring out how to make the protein and beginning to study it. And to make the protein, they ended up relying on a expression vector provided by my neighbor here at Vanderbilt and good friend, Rich Breyer, which turned out we still use that as the basis for expressing this protein in E. coli. And so that was sort of the early work on the project. Later on, Masayoshi Sakakura came in and did just amazing NMR work in which he showed that under NMR conditions, there's an equilibrium between a fully folded form of the protein and a partially folded form where the first transmembrane segment is dissociated from the rest of the protein and the other 3 transmembrane segments form a molten globular-like state. To this day, no one's ever been able to characterize the unfolding, misfolding of a membrane protein like Masayoshi did in this study. Really just a wonderful work. So what we set out to do, though, was to test the hypothesis. You have these mutations in PMP22 that cause a disease, Charcot-Marie-Tooth disease. Our hypothesis is that what these mutations do is they destabilize the membrane protein, making it prone to misfold under physiological conditions, leading to mistrafficking and loss of function. So we set out to test that hypothesis, and the person who did finish the test of the hypothesis was Jonathan Schleyback, who's now on the faculty at Indiana University and who's coming to biochemistry to give a talk next year. And so I won't go through this data in any sort of gory detail, but Jonathan developed not only a way of quantitating the stability of PMP22, which was the first time that had ever been done for any eukaryotic membrane protein except for glycophorin A. Not only did he do that, but he developed a quantitative mammalian cell trafficking assay for PMP22, which was the first mammalian cell biology experiments ever done in my lab. And that was all John. And so he tested the hypothesis, and the data is shown here below. What Jonathan found is there's a linear relationship between the stability of the protein and the trafficking of the protein. There's a linear relationship between the trafficking of the protein and and health, nerve health. So if you have an unstable mutant, it mistraffics and it causes low nerve conduction velocity, which is one of the key symptoms of Charcot-Marie-Tooth disease. And here is the relationship between stability and nerve conduction velocity. Unstable mutants cause disease. And so this was Jonathan's work establishing the basis for type E Charcot-Marie-Tooth disease. And this work was published in JACS. And let me just editorialize very briefly here about impact of papers. So I've published about 175 papers, and I assess that this is one of my top 5 papers ever, right? And yet this paper is cited number 78 out of 175 of my papers. And so I have no idea why this paper has not been cited more heavily, even though to me it's very clear this was one of the most important papers of my career. So I sort of take the view, if you have a highly cited paper, it's probably a really good paper. I agree with that. But if your paper is not highly cited, it means almost absolutely nothing. There could be any one of a number of reasons why a really fantastic paper doesn't get highly cited. And so anyways, that's just my opinion, but that's my opinion. So now Jeffrey Lee, who's a current postdoc in the lab, has followed up on this work by doing NMR studies of these different disease mutant forms. And what Jeffrey has shown is that all these mutant forms, when they're folded, fold in a similar manner to the wild type protein. And when they unfold, the unfolded state is similar in structure to the unfolded state of the wild type protein. So the real defect underlying this disease is simply the position of the equilibrium between the folded form and the unfolded form of PNP22. So we've continued a number of other studies of PNP22. This is the team that's pursued this. We have both collaborators and people from my lab, students from other labs. I especially want to note Justin Marinko, who's been studied the folding of the wild type protein, which also can cause Charcot-Marie-Tooth disease when it gets overexpressed. And so Justin has shown that if you overexpress PNP22 in mammalian cells, that also causes misfolding and mistrafficking. And our collaborator on that project is Bruce Carter, who's doing a lot of the cell biological work related to understanding exactly why does this take place. And again, we have a number of other wonderful collaborators. I'm not going to go through all the details here. I do want to note that Kate Mittendorf and Melanie Ohi did some beautiful studies where they found that if you reconstitute purified PNP22 into lipid bilayers, that they don't form traditional vesicles. What they form instead, and this is shown using cryo-EM and other types of EM, you get a central vesicle that gets wrapped around by flattened vesicles. This is very, very similar organization to what's seen in myelin. And so it really strongly suggests that PNP22, whose function has never really been well understood, probably plays a structural role in myelin among other functions. So, at this point in time, we feel like we are sort of experts at the folding and misfolding of human membrane proteins as it relates to health and disease. And we published this 70-page review that has like 950 references in it. And this review, published in 2019, has a whole section dedicated to the endoplasmic reticulum. And so hearkening back to the title of this talk. Now, one last point about PNP22 is our years of basic science studies of this protein have led now to an early stage drug discovery project involving members of Bruce Carter's lab, members of my lab, and then the Deerfield Investment Group's pharmaceutical company, 3DC. And this is mediated through a Vanderbilt Deerfield collaboration known as Ancora. And I want to point out another mentor of mine, and that is Dr. Margaret Reed of our CTTC office. Margaret is the interface between the Vanderbilt part of this project and the Deerfield 3DC part of this project. And she steered us through a very complicated proposal process to get funded through this mechanism. And she's just been a wonderful colleague and friend and mentor over the last couple of years. And this is a Vestigo photograph that I kind of liked. I'll tell you who everyone else is here later. But basically now we're pursuing early stage drug development for Charcot-Marie-Tooth disease based on the basic science results that we've generated over the last 20 years on this project. And it's really a lot of fun. It's a new direction for the lab. And in fact, if you look at the current version of my lab depicted here, about half the people in this picture are doing something related to high throughput screening in work that ultimately is designed to hopefully find therapeutics. And I want to acknowledge that Josh Bauer in Vanderbilt's high throughput screening core is now sort of an honorary member of the lab because he sees so much of us. So that's a picture taken this summer during that very brief time when we were allowed to even get rid of our masks for a while before the Delta variant came roaring through again. So I mentioned to you that I would take you all the way through one project in the lab and then back up and mention several other projects very briefly. I'm at that point now, and so that's what we're going to do. So another project that we had was a G protein coupled receptor project, and this was from about 2005 to 2015. And this turned out to be a project that had its challenges, and eventually we phased out this project. However, before we phased it out, we got what I thought was one really fantastic paper on this work, and this reflected the work of a postdoc in my lab, Tiandi Zhang, and a student in Tina Iverson and Sarah Sevagurovich's lab, Chu Yan Shen. And so what they were able to do was do studies of NMR studies of an arrestin while it was actually bound to rhodopsin in bicelles. So this was an enormous protein-protein membrane-bound complex. And the fact that we were able to get really interesting results out of this was really, I thought, a wonderful achievement. And we were especially thrilled with this PNAS paper because it turned out when the paper got accepted and we found out who the editor was none other than Brian Kobilka. And this was about 2 months after he got his Nobel Prize in chemistry was announced. So that was a really sort of a special paper. Now, there were sort of two things that happened that led to the GPCR project eventually being phased out. And one was that a key person on the project was my grad student, Anne Carpe. And Anne was really a wonderful, wonderful student. And so, and she was also a wonderful, wonderful person. And as many of you know, and very, very sadly, Anne passed away from cancer in 2008. And she really was the intellectual driver of my lab's part of the project. And you can sort of get that from the picture here. She just had a zest for science that was irrepressible. And so that was a really big blow to the project when she was no longer able to work on it. I also concluded that there are projects in science, areas of study where if you really want to make much progress, you have to devote your whole lab to it. And I came to the conclusion that that's the way G protein coupled receptors are. And I was not willing to reconfigure my entire world to be based on studying GPCRs. So we eventually phased out this project, but we're very happy and enjoyed the project while it lasted and enjoyed our interactions with our collaborators and trainees. So another project then was to begin work on studying how mutations in the casein Q1 potassium channel cause long QT syndrome and other arrhythmias. And so this project is ongoing to this day. And it really started off as a collaboration with Al George when he was here at Vanderbilt. And Carlos Vanuet, excuse me. Carlos is his lieutenant. And very quickly, then we also, Jens Myler became a collaborator on this project, long-term collaborator. And Jared Smith has done a lot of work on this project with us over the years as well. Wonderful collaborator. And then the folks who really got this project started in the lab, all four of these folks are now in faculty equivalent positions. And so this has been a really sprawling project. Here are some of the other people that have been working on this project. So this is Keenan Taylor. So Keenan determined the NMR structure of the intermediate state of the voltage sensor of KCNQ1. It was a marvelous technical accomplishment. Did a great job. Dumgung Peng started the project. Gary Lorgan did a sabbatical in my lab and then took up this protein. Has been a wonderful collaborator over the years. Cheryl Law studied one of the regulatory proteins for the project. I love this picture of Georg Kunze because he looks so serious. And Georg is a man of very few words. But I realized when I saw this picture, the thing that's wrong with this picture is Georg always has a smile on his face. And to see him actually being serious here is interesting. Now, this is Brett Kroenke. And Brett was a postdoc in the lab, and he's now in the faculty at BUMC. This was Brett's proudest moment in the lab. We went on a hike, and we came to this bridge that had been washed out and was underwater. And there was a sign up that said it was the Troll Bridge. And so we figured this was the end of the hike. We just have to turn back and go back. But Brett immediately sloshed into that filthy water and repaired the Troll Bridge so that we could cross over and continue our hike. And I thought, it kind of said something about Brett's personality that really is true, just a very positive, don't look back sort of guy. So now Hui Huang did a lot of beautiful work on KCNQ1. One of the things that she very recently did was found a mutation that causes super trafficking of the channel and atrial fibrillation. To my knowledge, the discovery of a super trafficking mutation that causes disease is the discovery of a novel disease mechanism. We also looked at 50 different mutant forms of long QT mutant forms of the channel and found that the most common cause of long QT syndrome are mutations that destabilize the channel, leading to its misfolding and mistrafficking. The exact same paradigm that we saw for PMP22. So this established a second disease that, in this case, we established this important relationship of, and what we believe is a very common relationship between human disease and membrane protein misfolding. Another project that's actually currently on hiatus, but probably not completely finished, is a collaboration with the lab of Roy Zent and Ambrapazzi studying integrin receptors. And this was carried out by a trio of postdocs, Zinwei, John, and Sijo. And what we were looking at here was how does the activation of the beta-1 integrins, which are the most common integrins involved in organizing organ development, for example, how does this differ from the well-characterized beta-3 based platelet integrin? And so this team of investigators basically showed that the beta-1 integrins are very different from the platelet integrins, which was very, very surprising. Got a very nice paper out of this. And so this project will probably continue, although we aren't working on it just at the moment. Marcus Vuhler is our NMR manager here at Vanderbilt. I just want to acknowledge that on almost all our projects, Marcus has a role in training my students and helping them run NMR. Finally, one last project is our amyloid precursor protein project, which is related to Alzheimer's disease. And so this project was started by Chuck Mobley and Hawk Jin Kim. Excuse me. And so they started this project back in the early 2000s. This is Anne after she had been through chemotherapy, and Chuck Mobley baked an enormous cake for her. Chuck liked to do everything big, and Chuck was a really, really good friend to Anne. Anyways, they started the project, and then it was really brought to fruition by Yuan Li Song, Paul Barrett, and undergraduate Andrew Beal, who was valedictorian this year. That's why he got to carry the flag. And so they determined the structure of the amyloid precursor protein transmembrane domain. And they also showed that this protein forms a stoichiometric complex with cholesterol that probably somehow promotes Alzheimer's disease. And in years since then, we've been trying to figure out how it does that. Other people have come on to the amyloid precursor protein project, including our dear collaborator, Ann Kenworthy, who's moved to University of Virginia, but we continue to collaborate with. Catherine Dieterich did a beautiful study comparing the structure of the amyloid precursor protein to the Notch transmembrane protein. Rob McFeeder did a sabbatical in my lab and has provided a lot of collaboration and on and on. And Lauren LaPointe is now at FDA. Matt Castro is still in the lab, finishing his PhD studies. This is his model of a nanodisk, another type of model membrane. So anyway, so that's the team for that. And let me just move to my closing comments here. So probably the best day ever for the Sanders Lab was the Eclipse picnic in 2017, when there was a total eclipse here in Middle Tennessee. We drove just north of Nashville and had a picnic, just us and our 80 closest friends there. And this photo was taken about 20 minutes before total eclipse. John York, who I consider to be a mentor, and my former chairperson, now at Impossible Foods, was at the picnic. That was a really, really fun day. Seeing a total eclipse, as any of you know who have seen it, it's a euphoric type of experience. Another fun day for the lab was when we all put on our togas and went over to the Parthenon located across the street here at Vanderbilt University. And that explains Arena and Becky's outfits. This is the extended lab photo where we have both the lab members, but also members of their family and their partners. That was a really fun day. And let me just conclude then with a couple last words about mentoring. So this is a couple of my students, Kate and Catherine, and their moment of glory when they graduated. And the photo here on the left, so this is Dean Larry Marnett, the Dean of Basic Science here in the School of Medicine Basic Sciences. And this is Mike Goodman. So Mike was one of Richard Armstrong's final students, and when Richard passed away, then Mike actually went to work under Larry's guidance. So Larry mentored him. And I personally am mentored by Larry on an almost daily basis as an associate dean. He's just a remarkably gifted mentor, and I continue to learn stuff. One of the things that Larry occasionally will say to me after a really bad meeting is he'll say, well, we're never going to get that hour back again. And I would just like to make one last observation about mentoring, that when you mentor someone, you're never, ever going to say that about mentoring, that it was a waste of time. You will always be glad for the time you spent mentoring someone else and trying to help them to get to the next step of whatever it is they're trying to achieve. And with that, I'm going to close. And thank you all for attending and for listening. And I hope you all have a great day and a great weekend.
Thank you, Chuck. There's never a good way to end these kind of Zoom things. But that was a wonderful talk, and thank you for sharing with us.
My pleasure. And again, everyone, take care. And it's one of these over and out moments. If anyone would like a recording, just shoot me an email, and there's the link if you want to download the slides. So bye-bye, everyone.
Thanks, Chuck, and congratulations.
Bye. Congratulations.